So the title of my talk is an unbiased cross-validation of cryo-EM maps. So I'm going to start with the motivation of our work, why it's important to validate cryo-EM maps, the reasons behind it. Then I'm going to explain to you the protocol and the main idea of our work, which is to use an independent cross-validation set, a control validation set that is not going to be used in the refinement for validating the cryo-EM maps. Then I will present to you the BYN Probability, which is a method that we've been developing to calculate the probability of a three-dimensional structure given a set of particle images. Then I will present two tests that monitor the quality of cryo-EM maps over the control set, which is the Cumulative Log Posterior and the similarity between the probability distributions of the cryo-EM maps. I'm going to explain it to you further on. So the motivation of our work. So as we saw in the morning, Cryo-EM has revolutionized structural biology. You can now find Cryo-EM maps at less than 2 Armstrong, and this is really amazing. And basically, Cryo-EM is the structural biology technique of the moment, and it will be for many decades to go. So in Cryo-EM, the gold standard approach is typically used to generate the refinement. So what you do is you take your particle images and you randomly select two sets. And you build the three-dimensional models independently and using an expectation maximization algorithm. So at each round of iteration, what you do is you compare these two maps using the Fourier shell correlation. So basically the correlation in Fourier space, and a fixed threshold is used to determine the resolution of your system, and then this process is iterated. However, cryo-EM maps may suffer from overfitting. And some reasons are that the resolution estimate depends on the symmetry of your molecule, on the Fourier shell radius that you use, on the threshold that you use, on the mask, and on the orthogonality of the signal and the noise. So there are many assumptions that might influence your resolution estimate. And so some possible issues. For example, in the recent map challenge, it was shown that there is no absolute gold standard, that there are user-dependent workflows, and that there can be over-manipulation of your data in your maps. There have been found inconsistencies between the map, the resolution reported in the map and the resolution reported in the model for about 30% of the reported data. And also, this has been typically known in this community that maps can be reconstructed from pure noise particles. So there have been many methods that have been used to validate the cryo-EM maps. So, for example, randomization of the phases beyond a certain frequency can give you signs of overfitting in the Fourier shell correlation curve. There have been proposed reference-free pipelines that use non-fixed thresholds for the FFC curves. There have been methods that propose to measure the local resolution instead of an absolute resolution of your map. Also, for example, the predictability of the particle alignment can give you indications of the quality of your maps, and there have been many more methods. However, a main disadvantage of the current approaches is that they rely on averaged or manipulated data, which are maps and models. And so what they do is they discard the raw data, the pure particle images that contain information that shouldn't be averaged or processed, right? That you could use to validate your maps. So this is the idea of our work. It's to use a small control set, a control set of particles that are completely omitted from the refinement process, And this would give us an unbiased estimator of the quality of the reconstructions. So this is based on machine learning approaches where you have a test set, a test data set that you don't use to create your models, or on the R-free in X-ray crystallography where you completely omit a data set from your refinement. So now I want to explain to you our protocol or idea. So we want to keep the gold standard refinement. So you have your two sets, and at each iteration process, they're completely independent. You have your two reconstructions, and you compare them, and you do all this normal refinement process. But at the same time, you have your control set of particles. And this is an independent set that is not used in the refinement process. And so what we do is, at each iteration step, we take the two maps generated, and then we low-pass filter them. So basically, we Fourier transform them, we cut at a certain frequency, and we generate these filtered, low-pass filtered maps. And what we do is we calculate the probabilities of these maps given the control set. So what is the probability of the system, of these maps, when I add higher frequencies to them, when I am in a different refinement iteration process? And basically, we're going to ask two questions. So the first question is, how probable are these maps? So do they become more probable as the refinement increases? Do they become more probable as we add higher frequencies to the maps? And the second question that we're going to ask is, how similar are these maps? So normally what you do with the Fourier shell correlation is you compare these two maps in Fourier space. So now what we're going to do is we're going to compare the probability distributions of the maps generated from set 1 and set 2. And I'm going to explain to you further on what these consist of. So first I want to talk to you about the BYN probability. So this is a work that we've been developing with Gerhard Hummer at the NIH and at the Max Planck Institute. And the idea is to use Bayesian analysis. So it's Bayesian inference of electron microscopy. And basically, it's to calculate the probability of a map given a particle image. So what it does is, your probability is the integral over all nuisance parameters that affect the experiment, the orientations, the defocus, the particle center, of this likelihood function times the prior probabilities of the map and the parameters. And so the likelihood function, it's basically a forward calculation where you start from your map, your three-dimensional structure, and then you have all these parameters that generated a calculated image from your map. And what you do is we assume Gaussian noise, or we have a very simple model of the noise, and it's the exponential of the cross correlation between the calculated image from your map and the observed particle image. And so basically, this is a measure of how similar is the calculated image to the observed image, given all this set of parameters. So because we do Bayesian analysis, we're going to integrate over all the parameter sets. So we're going to explore all of these different parameters. And at the end, we're going to have a number, a probability of this map. So for multiple images, so this was for one model, one map, and a particle image. So for multiple images, the posterior probability is simply the product of all the individual probabilities of those images. So this is like the cumulative log posterior. So basically, it would be the sum of the logarithm of the posterior probabilities. And what we have found is that this is a very good tool for model comparison. So if I have, for example, two different maps or two different models, and I use this BYN posterior, I can correctly discriminate and rank which model is more probable or which map is more probable. So we started developing this tool thinking about Cryo-EM of flexible systems. Cryo-EM of intrinsically disordered systems. And so basically the main idea is to do ensemble refinements and to combine these measures with simulations. So you have an ensemble of structures from your simulations, and what you want to do is you want to reweigh them, reweigh the ensemble so it matches your data best. We have also used this for determining the stoichiometry of proteins embedded in large super complexes where you have low resolution structures and you can't know how many copies of the proteins of your system are. But as an overall conclusion, basically, this method allows us to assign probabilities to a three-dimensional conformation, a three-dimensional structure, given a set of images. So now what we want to use it is for cross-validation. So we calculate these BYN probabilities over the control set to validate the Cryo-EM maps. So we use several benchmark systems. These are available at Empire, and they have different ranges of resolution from 5 to 3 Armstrong. And what we did was we took the raw particles available in this data set, and we omitted 5,000 particles from the refinement. And we did the normal Cryo-EM refinement using Reliant 3D refinement. So basically, the same thing that the authors reported with the symmetries and all the suggestions that they used, but omitting 5,000 particles from the refinement process. And we also had 2 sets that mimic overfitting. So we have the HIV one envelope trimer. There has been some polemic with this system where it has been suggested that that the resolution estimate and it has been overfitted. And we also have a set of pure noise images with the RAG1 and RAG2 complexes. So basically, instead of having the experimental images, the particle images, what we have is we have a set of images that only have Gaussian noise on them. So the first test that we want to ask is the cumulative log posterior. So we have the standard Cryo-EM refinement, and what we do is at each iteration step, we low-pass filter the maps. So we have maps at low frequency, which are low resolution, and maps that we keep adding higher frequencies to them, increasing the resolution of your system. And so the first question that we ask ourselves is, how probable are these maps? And so here, what I'm graphing is the cumulative log posterior. So basically, the sum of the posterior probabilities over the control set of the particles as a function of the frequency cutoff of the low-pass filtering. So here, what you see is in maroon is the first iteration, and in green is the last iteration. And the solid is the data for the maps from set 1, and the dashed line is the data for the sets 2. And as this number increases, more probable are these maps. So here we find 3 important results. The first thing is that the cumulative probability increases as a function of the frequency cutoff. So as I'm adding higher frequencies to my map, there's an increase of the posterior probability up to a certain plateau. The second result that we find is that the cumulative probability increases as a function of the iteration. So one would expect that as I refine more my map, as I make my map better in the refinement process, the information from the control set tells you an indication that you're doing a good job, that your refinement is indeed making your map better. And this is completely unbiased because the particles in this set are not used for the refinement. The third result that we find here is that there is very small difference between the results from set 1 and from set 2. And this is kind of what is expected in these refinement algorithms in the gold standard, where you divide your set into two sets and you compare the two maps. Those maps are very similar. So we did these for the standard Cryo-EM systems, and we find a very similar behavior. An increase of the posterior probability as a function of the frequency cutoff. For the different refinement iterations, we see an increase of the posterior probability. However, for the overfitted data sets, for the noise particles or the HIV enveloped tremors, what we find is a decrease in the posterior probability, or a slight decrease in the posterior probability, or that there is no effect when I add higher frequencies to my map. So basically, this would be the first test that one should do to monitor the quality of your Cryo-EM maps. You have to check that your posterior probability increases as a function of the iteration and as a function of the frequency added to these maps. So now I want to explain the second test, which is the similarity between the posterior distributions over the control set. So we have exactly the same methodology at each iteration. We low-pass filter the maps. And now we want to ask ourselves, how similar are the two maps, but given the posterior probability distribution? So here, what I'm graphing is, for one iteration, the distribution of the log posterior over the control set for a low frequency for set 1, which is the black line, for set 2, which is the red line, and for a higher frequency for set 1 and for set 2. So as you can see, the red and the black line are very similar, and the blue and the cyan line are also very similar, which is expected from the refinement algorithm. You expect that when you refine and you have these two sets, that your maps are similar. However, if you look at the difference in these posterior distributions, we find that the maps with higher frequency, that have more frequencies added, have a higher differences in the posterior probability distributions. So basically, qualitatively, we find that the difference between the distributions is larger for higher frequencies. And if you think about this, this is similar to what happens in the Fourier share correlation. So at low frequencies, your correlation is almost 1, because your maps are very similar at low frequencies. And you start adding higher frequencies to your maps, and then the correlation starts going down. And this is what we see here. So to quantify this, we use a normalized Janssen-Shannon divergence. This is a Kullback-Leibler divergence symmetrized to compare the similarity between probability distributions. And we normalize it by the Shannon entropies, so it doesn't depend on the number of particles of your system. So basically, what we're graphing here is this normalized Janssen-Shannon as a function of the frequency cutoff. And what this indicates is, as this goes higher, the probability distributions are less similar. So here, what we find are two things. The first thing is the confirmation that as we add higher frequencies to our maps, less similar are these probability distributions. And also, as the iteration step increases, we see that this NJST reaches a plateau at higher frequencies. So basically, there's a plateau. There's a frequency plateau that's reached by the first iteration, which is lower than for the greater iteration. So as my iteration increases, this slope changes. So we find a very similar behavior for the standard cryo-EM systems. So basically, as higher frequencies are added to these maps, we see that the normalized Janssen-Shannon divergence increases, so the probability distributions are less similar. And we also see that the plateau where the plateau that the data reaches as a function of the iteration increases with the iteration. So for the overfitted system, we find a very different behavior. We find a random-like behavior or a very plateau-like behavior where you don't see really an increase of this similarity measure as a function of the frequency cutoff that we use. So the conclusion is that the distributions should become less similar with higher frequency cutoffs. So finally, we realized that we could fit this normalized Janssen-Shannon divergence data with an inverse exponential function of this form. And basically, this gamma in the exponent gives us an indication of where the data plateaus. So for higher iterations, we find that the plateau of these exponential fits is different and increases. And so what we did was we compared this to the map resolution obtained in the standard way using the gold standard. And so here, what I'm graphing is, this 1 point represents one iteration. So the value on the x-axis is 1 over the resolution obtained with the Fourier shell correlation and the 0.143 threshold. And here on the y-axis, what I'm graphing is this new decay frequency that I used to fit the data from the control set. The amazing thing is that we obtain very high correlations of the order of 0.9. And basically, the information of the map resolution is contained in this control set, in this particle control set. So how I want to conclude is that even for a small particle set, it's possible to extract information about the map resolution in an unbiased manner. So we've been working a lot on developing the code. So we have the BYN code with a GPU acceleration available at GitHub. And we also now have developed a cross-validation tutorial that gives you a step-by-step tutorial on how to implement these cross-validation steps. So the conclusion of our work is very simple, and we think that a controlled particle set not used to generate the reconstructions can allow for an unbiased assessment of overfitting in Cryo-EM. So lastly, what I want to do is I want to thank my student, Sebastian, which is the one that has done all the work, my collaborators, and you for your attention. Thank you very much.